أهلا ومرحبا بكل الدكاترة الحاضرين معنا والخبراء. أهلا ومرحبا بكم في هذا الملتقى الدولي، ملتقى الوراثة التطبيقية بعنوان Biodiversity, Health, Security and Food. إذا حالتم أهلا ومرحبا بكم. بداية في اليوم الثاني من هذا الملتقى يتفضل بروفيسور جوهر سهيل بإلقاء كلمة افتتاحية لهذا اليوم الثاني من الملتقى.
بسم الله والحمد لله والصلاة والسلام على رسول الله. إذا متابعة لما قمنا للحدث الذي ابتدأ البارحة، اليوم سيكون اليوم الثاني مواصلة لأعمالنا إن شاء الله. واليوم ستكون هناك محاضرات افتتاحية إن شاء الله، ومن بعد سيكون ورشات، وسنحدد أين ستكون الورشات، الحيوان والإنسان. مرحبا بكم جميعا، شكرا لتلبيتكم الدعوة. حلتم أهلا ونزلتم سهلا، وبارك الله فيكم على الاستجابة للدعوة. السلام عليكم.
And we will have the information soon. So we don't know.
So good morning everyone. Welcome to all our teachers, our doctorants, our students in the international seminar Biodiversity, Food Security and Health. It's truly an honor to have each of you in our seminar to bring your expertise for advancing knowledge in biodiversity and genetics. Let's make the most of this opportunity to exchange ideas, collaborate and push the boundaries of scientific discovery. So welcome to all. First, I invite Ms. BROCKMANN to present his topic about genetics information for the maintenance and improvement of small breeds like German black-eared cattle.
Can I take this micro? I like this more. Good morning, everybody. First of all, I'd like to thank the organizers for inviting me and to present data of our research here. I'm going to talk about the German black-pied cattle, which is an endangered breed, one of the many we have worldwide. And I'll give you an example what we did to contribute for their protection. So I come from Germany, from Berlin, and these black-pied cattle once contributed to the high-yielding Holstein cattle. Worldwide, Holstein gives the highest amount of milk. So originally, the black-pied cattle came from the area around the North Sea. This is what you see here on the right side. Germany is is dark blue, and the North Sea is in the northern part of Europe. And there we had these black-pied cattle already 200 years ago, and they were quite good in milk yield, but this was a dual-purpose cattle. And then 200 years ago, during the big famine, so many Germans moved out to Northern America, to USA and Canada, and they took with them these cattle. So, but what happened then? In America, they selected the individuals for high milk yield, for very high milk yield only, and this was the reason why we had the Holsteins later on. And so therefore, the Holsteins were re-imported back to Europe in the 60s and 70s of the last century. But this led to the replacement of the DSN in a very short time. Only 10 years later, there were only about 4,000 DSN cattle in Germany from 1 million we had before. So, and this was the reason to found an association called Gene Reserve DSN. Last year, there was a 50th anniversary of this breeding organization, and last year we had only 2,500 cattle which were registered in the herd book. So this means it's really an endangered and small breed. The DSN have some advantages, and the major disadvantage is the low milk yield. The average milk yield is about 7,200 kilograms per lactation. This seems to be quite high, but it's low compared to Holsteins, which have about 2,800 kilos more, and milk makes the money. And because of the low milk yield, we have only very few breeders who still keep the DSN. And therefore, it is a very small population. But therefore, also the government supports the breeders financially. Per purebred cow, the breeder gets about 100 euros per year. There are, of course, some advantages of DSN, because the milk quality is really good. The contents of fat and protein are very high compared to Holstein in particular. It's a dual-purpose breed, so we have, in addition to milk, we have also the beef, but you can't get money with the beef. And we also know the fertility is very high, the individuals are very robust, and the feed efficiency is very good. So we started with the research about 10 years ago, and the goal, not only from the breeders' organization, but also for us, is to minimize the inbreeding level, to maintain the high diversity, and to improve the positive traits, in particular also find markers, find genetic markers for the improvement of milk, because milk makes money, and this would lead in the future to a lower support of the government. So what did we do? We collected blood or tissue from the ear tags when the individuals get their individual label, and did genotyping. For genotyping, we used the commercial chips, SNP chips, which is done a lot nowadays. But then we also had the funding money for whole genome sequencing of about 300 individuals. And I said the population size is 2,500. So we sequenced about 1/10 of the whole population. And so by this way, we covered almost the entire diversity, genetic diversity of the whole population. And in addition to the whole genome sequencing, we have now in our hands also long-read sequencing, which improves the quality of the sequencing data we have in our hands. So with all this data of about 4,500 individuals, we continued our research. So I already mentioned that we use the commercial SNP chips, which is quite good for paternity testing and some other things. But if you really want to know the special things of DSN, you also have to have the access to the specific mutations in DSN. So, and therefore, we developed a very specific DSN chip. And this DSN chip contained genetic information which is important for the breed. We got the information from our 300 whole genome sequenced individuals. So what kind of SNPs did we take? We had space for about 200,000 SNPs on the chip, which is quite a good number. First, we took all the SNPs which are informative from the commercial SNP chips, which were about 34,000. And then we took all the information we had already in our hands from genome-wide association studies, where we knew SNPs are associated with milk yield, with fertility, or with some health traits. We added all the information which we obtained from in silico analysis of the whole genome sequencing data for high functional impact SNPs. So also SNPs which would change the proteins, the quality, or also the amount of proteins. Then we added DSN-specific SNPs, which are between genes, and we know these might be very important for the gene regulation. And then we added 5 different categories which are important for paternity testing, for checking the purity, and for filling gaps to have covered the whole genome in a very fine scale. Using this information, we checked how good our DSN-specific SNP chip is. So, and here we check not only DSN. DSN is the first cattle here on the left side, on the upper row. And our chip worked very well. 87% of all the SNPs we selected really worked, which was really good. But we also found for several other breeds we just tested, it works very well. And even one Bos indicus breed, namely Butana, was informative. So still half of the SNPs worked for Butana. So then we knew our chip is really good. So and we continued to genotype about 1,800 individuals with this chip. And this chip helped us to use the commercial chip, put in the information from this DSN chip later on, and also calculate the whole genome sequence of all our individuals for the whole genome and for all our cattle. So one of the questions we wanted to answer is, which breeds are highly related to DSN? Why did we want to answer this question? Because the highly related breeds could be a resource for selection of putative breeding bulls to improve the diversity in DSN, to bring in new blood. So we had access to our own 300 whole genome sequenced cattle, and then we took all the information from the 1,000 bull genomes project, where we found at that time 68 different Bos taurus breeds, and there were more than 1,000 individuals belonging to these 68 breeds. So doing the relationship analysis, we constructed the phylogenetic tree, and as you can see here, there were 4 clear clusters. So the breeds in the green cluster also contained DSN. This is where there is the red arrow. And the green cluster here on the right side, you can see is in the northern part of Europe. Here. Then we have the violet cluster of the breeds in the middle of Europe, and the blue cluster, which with breeds coming from the southern part and East Europe. Let's have a closer look to the green cluster here. And here we see DSN next to Dutch Frisian Red, Dutch Belted. These are the breeds you can see here in the middle with the picture. And these are also dual purpose breeds. So what is the main information from this analysis? First, DSN is definitely a separate breed from Holstein. So we can separate the individuals very clearly. And one of the goals of the DSN breeders is to have less than 10% of the genome of Holstein in DSN. Only then we would say these are purebred individuals, and only then the breeder would get money from the government. So this is one information. And the second information is that, for example, Dutch Frisian Red is very close to DSN, and this population could be used to identify potential bulls for improving the diversity of DSN. So we checked the pure breeding and diversity of DSN compared to Holstein. Because of the very small population size, 2,500 individuals only, it is important to have a low inbreeding level and to maintain the diversity we have. So for the genomic inbreeding, we examined the excess of homozygosity and the inbreeding coefficients. And we could see for all values we obtained here, the values for DSN here on the left, oops, are much smaller than the values for Holstein. And with respect to the nucleotide diversity and the observed heterozygosity, as measures for diversity, we can see that DSN is higher diverse than even Holstein. This means the breeders' organizations did a really great job during the past tens of years, which was really great. So I'll skip this. So we went on to do genome-wide association studies. Our major goal was to identify markers to improve milk yield, because this makes the money. So the population size we used for this genome-wide association studies was about 2,000 cows, because not all genotype cows had phenotypes already. We genotyped the calves, that means at a very young age, and lactation starts, of course, much later. So, and we used all the cows which had at least 3 completed lactations. And then we imputed the genetic information from the SNP chip level to the whole genome sequence level, and therefore we had 11 million or almost 12 million SNPs for each individual, which is very high. And I still want to mention this is DSN-specific information, which was really good and which helped us a lot to find significant markers. So we were particularly interested in milk traits, and then we used the genome-wide association study. That means the test statistics, where we put in the genetics, we put in the phenotypes, we considered genetic relationships, and we considered all the environmental effects that have that influence the milk yield. So this is the result of the genome-wide association study for milk yield, the so-called Manhattan plot. And here we see the different chromosomes and the test statistics. And you probably also see the red skyscrapers. And these are the few very significant and important results. So for milk yield, we see such significant findings on chromosomes 1, 3, and 8. So, and then we follow up these hits with the whole genome sequencing data. So this is a zoom in of the chromosome 8 hit we have here. And under the very significant SNPs which show us there is a gene contributing to milk yield, we found TLE1 and TLE1 and TLE4, and these are transcriptional co-regulators which are known from other studies, in particular in humans, but also in model animals. They contribute to the Wnt signaling, and Wnt signaling has to do with tissue differentiation and development. And so it was found that they contribute to the normal regulation of the mammary gland. And there's also evidence that these genes contribute to the development of mammary gland cancer. So this means these genes probably contribute to a normal and very good development of the mammary gland where milk is produced, which is very important. So another surprise was that another gene that you can find everywhere, and which is very well known for all the dairy cattle breeders, this is DGAT1, which is written here on top. DGAT1 is a gene on chromosome 14, and usually you find a hit on chromosome 14. But here on chromosome 14, there's nothing. And this means, albeit in Holstein, DGAT1 is a major gene contributing to milk yield, it doesn't play any role in DSN, albeit these two breeds are very close related. So this was a very interesting and very significant finding. So these are the results of the genome-wide association study for milk fat content. And as you can see here, there's a very high hit, a highly significant locus on chromosome 5. And this was really a surprise because we didn't see it when we started our research with the commercial chip. Because the commercial chip didn't contain the SNPs, which are important for DSN. Only after having the DSN-specific chip and the whole genome sequencing data, we found this significant gene, which is MGST1. This is also a gene which had been found in other breeds before, contributing to milk fat, but not at this effect size. The effects are much smaller in other breeds. MGST1 is a microsomal glutathione S transferase 1, and this contributes to the synthesis, secretion of lipids, and mitochondrial activity, glycolytic processes, which is all associated with the production of milk fat, which makes sense. So we know now the frequency, and we can use the beneficial allele to select for higher fat content in the milk of DSN. And finally, I also want to show you a result of the genome-wide association study for milk protein content. And here are also findings on chromosome 3, 6, and 10. So we looked up all these different regions on the different chromosomes, but there is one region on chromosome 6. So 6 is famous, and we know there are all the different proteins, milk proteins on the caseins. And so indeed, the highest significance here, the top SNP, is very close to alpha S1 casein. It's not in the protein. It's very likely in the regulatory region, which makes also sense, that the production of alpha S1 casein is regulated in this breed. And so the selection for the favorable allele could improve further the milk protein content in DSN milk. So this is just to show you, we also used the information we had for health traits. We found loci for the sensitivity, for clinical mastitis, the mammary gland disease, for endoparasite resistance, and also for fertility. For fertility, it was quite interesting because in the Holstein breed, there is one haplotype segregating. So, and this is risky. Breeders don't want this haplotype because it leads to a premature termination of pregnancy. So, but this haplotype does not exist in DSN, which is really a good information. So then was the question, and the breeders always ask us, you have all this information, so how can you help us? And this is really a challenge because the population is small. The breeders exactly know what they want. They want to improve the milk yield, but they also want to have the high diversity, and so on and so on. And then we thought the population is too small to really estimate genomic breeding values. The population is too small, so therefore the accuracy of breeding values is so bad that we can't give them genomic breeding values. But what we did is we took the most significant SNPs, only the most significant SNPs, and calculated gene score. So we genotyped the animals, and then we took the SNPs, which had the highest effects on milk yield or fat yield or protein yield, for example, and summed it up. Very easy. So we came up with gene scores for milk yield, and here are 10 individuals of the top, of the end of the tails, the best ones and the worst ones. So the green ones, the green animals, the 5 green animals have the highest milk yield, and the red ones have the lowest milk yield in the population. So this is, the best ones have more than 1,000 kilograms as the average of the population, and the bad ones are about 1,000 below the average. So, but then you also see the gene score for protein kilos, for protein percentage, fat kilo, and fat percentage, and then the breeder can decide what he really is interested in. So this is not a genetic value for the individual, it's just a helper which combines the information we have so far. So also, this can be used in addition to the breeder's selection they want to do. So, and then I want to sum up. We have a lot of genetic information, of genomic information for DSN. What can we do with this information? First, and this is also very important for the breeder, we can genotype all the calves in order to do the parentage testing, to check the purity of the breed, to check the inbreeding or the diversity. This is important for getting the governmental funding. Then, because we know the relationship to other breeds, we can give some recommendations of potential breeding bulls for the future. And then finally, we have basic information for the development of these genomic breeding values. We give a basic information with a gene score. And I want to stress basic information. It's not a genomic breeding value so far. What we want to do with all this information is we take the very best SNPs associated with different traits, and we want to bring it to the commercial chip, because there is still some space on these commercial chips, and there's an agreement between the breeders and the company who sells the chip. So there is still space. We want to bring these few thousand SNPs on the chip so that we can, at a very low amount of money, genotype each individual immediately. And such chips cost then about 25 euros only. And this is its worth. So finally, I want to say who did all the work. Of course, we had project funding in my group. And those people who contributed most are two postdocs and one PhD student who developed really the DSN-specific chip. We collaborated with another university in Germany. We collaborated with the breeding associations and with the breeders themselves. And this was always fun to meet the breeders. This was the most important thing here. So now I'm at the end of my contribution. I'm glad to take questions if we still have time.
Yes, yes, yes. You have time. Thank you, Professor BROCKMANN, for your very rich presentation. So to be fast, you can have one or two questions if you want, and after we pass to another presentation.
Can you use this? Can you use this?
Okay.
Thank you for the nice presentation. I learned a lot actually. I have actually a technical question when you mentioned the fact that you are using commercial chips to provide the genetic landscape of all these different species of cow. I wonder, so the issue here that I see is that when you want to bring back that to the, this new SNP into a new chip, you are going to design some probes that usually, depending the companies, the size of the probes will range from 25 to 70 base pair. And within this probe size, you need to make sure that you don't have any other mutations that will prevent your chip to not working right. So the question is that how you are, and you will need to have the whole genome sequencing for all these different kind of code to do that. So how are you going to be sure that your new SNP will be effective in the chip?
So first So if I understood everything correctly, you want to know how can we be sure that the SNPs we want to put on the chip are the right ones?
The probes will be effective, that you are not going to have other SNPs or other mutations within the probes.
Yeah. Yeah. First, we have the whole genome sequence information. So this helps us to design good probes.
So for each of the
there are of course some better ones, some which do not work as good. But we have these SNPs. Many of these SNPs are already on our DSN specific chip.
Okay.
Yeah. So and they work already very well because we did some quality checks beforehand before we designed the chip. So. We are pretty sure, and we are pretty sure that all these SNPs work well in the commercial chip because they are also on our chip already.
Okay.
Yeah.
Thanks.
Thank you. Next question, Professor.
Thank you, Gudrun, for the nice presentation. You know, I'm well known what you've done in DSN since I was in your lab several times since my PhD. But the new things I realized here that you calculate the gene score, which is the basic information to select the best animals. How many individuals you genotype to calculate gene score?
Well, the gene score is a value that we calculate for every individual. So we genotype all individuals. We genotype already every calf. So, and then we take the SNP effects that we know from our genome-wide association study, and we just sum them up, and this can be done for every individual.
Yes, but you choose the best 5 individuals?
No. So what we do then, so we have the gene score for every individual.
Yes.
Yeah? So then we sort the gene scores for the trait the breeder is interested in. In my presentation, we have sorted them for milk yield.
Yeah.
But we also can sort them for protein yield, for example. If the breeder says, we want to improve protein yield, yeah, then he has the information, these are my best individuals, and these are those I'm not so much interested in. And then he can say, okay, I wanted to use these and these individuals to breed them with these and these bulls. And among the individuals I have, I take those because they contribute to improve my protein yield.
So is the breeding company, if you come to the reality in the practice, if the breeding company use the gene score information instead of the breeding value in Germany?
No, not instead of.
Okay.
And this is what it's really important to understand this. The gene score is not a breeding value.
Yeah, it's clear. Yeah.
Because of the small populations, the effect size is not safe. Yeah? And therefore, it's an additional tool for making breeding decisions. Yeah?
Okay.
A helper tool.
Okay, thank you so much.
In the same context, are the gene score reflect genetic additive value?
Yes.
Yeah.
We did a correlation analysis. Yes. Yeah. And though it's quite good reflected, or quite good, the R is about 0.6. Yeah.
Thank you.
The last technical questions for the long read sequence. The sample size was 10, I think, yeah?
Yes.
So what you will do with this information, 10 individuals, and the whole population is 2,500, correct? So what's your information you will gain if you use only 10 individuals for long read sequence?
So the big advantage of the long reads is first to fill gaps, then to have the haplotypes. You exactly know which SNPs are on one chromosome, and this helps us to find eventually the causal mutations contributing to our QTLs. This is a very big advantage. And then we can compare haplotypes also with other breeds to see which linked mutations contribute to a phenotype, because I think sometimes we won't be able to distinguish single mutations and say this mutation is responsible for this and this trait, because the population is too small.
Thank you. Next question.
Thank you. Good morning, very nice talk. I'm curious about the DGAT1 gene. Have you looked at some similar genes having the same function? That's kind of the balance of the absence of expression of DGAT genes in the DSN. I'm just curious to know if you looked at other genes doing the same thing or having the same function.
I didn't get the first part of your question. So we didn't see DGAT1, but we did see other genes. But what was your question?
Do you have like a gene that balance for the loss or the absence of DGAT1?
Oh, DGAT1 is not absent. DGAT1 is not absent. So our data only show there is no variability in the gene.
Oh, so it's
everything is fixed. There is only one haplotype, and so therefore there is no possibility for selection. And in the DSN population, it's even the good haplotype is fixed. So we can't further improve it. Everything is already at maximum with respect to DGAT1.
Thank you.
The gene is not lost. Yeah.
Thank you.
Okay, just to complete this information, DGAT1 is present in the DSN.
Yeah.
They have a role in yield milk, but the DSN are a small population, so they have the same variability in DGAT1 that other genes make a role in production.
Yeah.
That is what you...
Also with respect to DGAT1, there is no variability in DSN. There's only one allele fixed. Only one allele.
They make a role in production.
Sure, it contributes because DGAT1 is important for the production of the triglycerides. Yeah? Without DGAT1, there are mutant mice and they do not produce milk. Yeah? DGAT1 is an essential enzyme in the production of lipids.
Okay, thank you.
Yeah.
Thank you.
My question. For DSM, in one of your slides, you recommend some crossbreeding with other breed which is near to DSM.
Yeah, this is not crossbreeding. So we had different pure breeds, yeah, and we identified breeds which are very similar in their whole genome.
Similar.
Yeah, similar. Yeah, it's not a crossbreed. They are similar, and therefore they are potential breeding mating partners to improve DSN without diluting DSN with a different breed.
So to give new blood to your population.
Yeah. Yeah. Yeah.
Thank you. Thank you all. Now we will pass to the number presentation. I need Mr. Pareil to put Mr. Olivier is not here, but he said
Could you read it?
You have to keep this.
Yeah.
So Mr. Paulo and Mr. Olivier are not here. So they sent the presentation on the video. On this presentation is part of our project Scalamidi, Prima Scalamidi.
So Paulo is the principal coordinator, international coordinator about our project Prima Section 1 called Scalami. So he can't come, so he sent us his presentation. We present it now. Scalamidi's project where we study sheep and chicken. We have technical problem with the presentation of Paulo, so we pass now to Professor Kahia to present her presentation about genetical laboratory.
So, Miss Kahia Samira presents his topic about genomic approach for biodiversity analysis.
واش مفورماتي هذا؟
جو سي با. نبقى نكلي ولا غادي نكلي؟ غادي نكلي؟
لا خصنا نمشيو مع 13.
ني با فورماتي هما.
دينا في كلي دينا في ولا كلي يو اس بي؟ لا 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 نديرو في بيسي واحد اخر. لا بيسي تاعك ما دير نطلع هذا مداري الا ما دارهاش ادابتاتور. لا ادابتاتور. دير بيسي تاعك. شوف الكلي تاعك.
ما فيهاش لي بور يو اس بي.
نو شغل ما حتاجش لي يو اس بي.
On me met des médailles.
اسكو سي بون فيديو. سي بون بعثت في الميل.
Il n'y a pas de connexion. Il faut faire partage de connexion pour.
Non, partage, il طلع connexion.
التليفون تاعي فضل. téléphone, il a dû le prendre en fils. téléphone, عندك mon téléphone. Je partage avec mon téléphone.
Je ne sais pas partager.
On s'excuse pour des raisons techniques. Maintenant, je pense, j'espère qu'on va passer maintenant à la présentation de Olivier Hanno.
هدية نتاعك؟ نتاعك هذا؟
C'est pas un Bluetooth. Faire partage de connexion. C'est partage de connexion.
هدية.
عندك partage de connexion.